Okay, so this is our batch review for long test number 1 for the 4th quarter. So I hope you have studied well. Take note of the following as we continue with our discussion. First of all, this would be the long test 1 guide. So there will be 40 points for our examination. And then the type of test, as you already know, multiple choice question, short essay, and provide what is asked for. And in terms of the breakdown of the question, we already mentioned this several times. We will be having 5 topics here: recombinant DNA technology, gel electrophoresis and polymerase chain reaction, Module 1.4, applications of DNA technology, Module 2.1, overview of photosynthesis, and Module 2.2, light-dependent reactions. And all of them will have 8 questions each for a total of 40 questions for the long test number 1. Now, in terms of our examination tomorrow, these are just a few of the reminders, very familiar already with all of us. First of all, you will answer on the answer sheet. So this time around, an answer sheet will indeed be provided. Already mentioned that we have MCQ, multiple choice question. So write the letters of the correct answer in the answer sheets. And also a short essay. If you're asked for 1 to 2 sentences, do write it that way. Or if you're asked for 2 to 3 sentences, do write it that way. And finally, you have provide what is being asked for. Okay, other reminders, you already know this. You can write or mark the questionnaire if you need to do so. And then you can make erasures if you don't have correction tape. So kung kakamali kayo, okay lang naman na burahin siya, make sure that you cross it out very clearly so that I will know what would be the correct answer that you have. And before I continue, just a reminder naman uli sa MCQ, multiple choice question, please make sure that you will be writing in capital letters rather than in small letters. Other reminders for the exam tomorrow is bring extra pen, no pencil please this time, and then go to the CR, get water before the examination starts, and finally put all your things on the walls of the classroom before we start with our exam. We only have 50 minutes, so make sure you are ready when I arrive so that we will be able to start immediately with our examination. Now, at the start of our quarter, we began talking about how humans determine plants and animal characteristics, and we are introduced to 2 kinds of doing so. Number 1 is what we call as classical breeding, and the other one is recombinant DNA technology. You already know from our previous discussion that the classical breeding takes too long because during the ancient time when the people noticed some very nice characteristics in the different organisms, they would mate together individuals that have the same characteristics, hoping that those characteristics will be shown or will manifest in the offspring. So later on, habang tumatagal ay nakita itong mga characteristics nito, pero masyadong matagal, or took a long time. But as compared to that, the recombinant DNA technology, the characteristics that you actually want to see in the organism that you have a focus on can immediately be placed by just simply taking the gene of interest from another organism and put it inside of that. So just like this flavor savor, wherein the characteristic that we want here, or the scientists wanted, is in order to have longer shelf life, they made use of recombinant DNA technology. So we talked about the classical breeding and the recombinant DNA technology, and we saw the advantages and disadvantages, and this led us to the talk, or to our discussion about what we call as the traditional biotechnology and also the new biotechnology, or what we call modern biotechnology. So the old is the traditional and the new is the modern biotechnology. So you already know this as well. So just remember that when we talk about traditional biotechnology, this is simple technique, usually a fermentation process, and we'll look at some examples aside from yogurt, cheese, preservation of pickles, and this is the use of natural ability of microorganisms. So this has been done in the past since ancient times. But when we talk about the modern biotechnology, this is just very, very recent, and make use of recombinant DNA technology. It is a new technique, and then there will be some modifications in the DNA, in the microbes, the plants, animals, and one example, production of insulin. So looking at that, the traditional biotechnology, it would be including as we mentioned a while ago, and this is the modern biotechnology like DNA profiling, genome analysis, and transgenesis. So here are some examples of the traditional biotechnology. So production of wine, production of beer, the cheese, dosa and curd, if you heard about it, particularly also vinegar, soy sauce, kimchi, because there's fermentation. So those would be some examples of what we call as traditional biotechnology. And you're familiar with the modern biotechnology. We already mentioned this: gene cloning, genetic engineering, recombinant DNA technology, human gene product, and GMOs like this one, the golden rice, and making use of gel electrophoresis. These are examples of modern biotechnology. So after talking about that, we talked about genetic engineering, recombinant DNA technology. So let's just compare this. When we say genetic engineering, please do remember that is simply altering the genetic makeup. And how do you alter? You insert, you remove, or you alter individual genes. So these are the three ways by which characteristics can be altered, characteristics can be added. So this is by inserting a DNA, a gene, by removing a gene, or by altering individual genes, changing them. Now, recombinant DNA technology, in this case, DNA sequence produced artificially by how? Joining pieces of DNA. So in this case, you are making use of techniques to cut and to paste different DNA fragments together. Now, our question might be right now, what's the difference between genetic engineering and recombinant DNA technology? Just remember that recombinant DNA technology is a very specific technique that is under genetic engineering. So genetic engineering is the umbrella term, the umbrella concept, and one of the techniques under genetic engineering will be recombinant DNA technology. And please do remember, the most important thing you have to remember for recombinant DNA technology is that there is the cutting and the pasting of different DNA fragments together. So let's look at that a little bit more. So in this case, what are the actual steps in genetic engineering? So there are 4 steps that we have to take note of. Number 1 is the breaking and joining of DNA molecules, so that we will be able to form recombinant DNA. So this means this will be DNA that comes from different sources, different species, and you put them together. The second one is finding a suitable gene carrier. Once you have your recombinant DNA, then you will need to put them together with a suitable gene carrier. And then third step would be the means of introducing the composite DNA. There will be it will mean nothing if you will not be able to put that in the actual organism that you want to change. So you have to have the means of introducing that. And number 4 is a method of selecting from a large population clones of recipient cells. So if you will be growing a lot of these cells, you must have a way by which you can select the ones that contains the recombinant DNA. Otherwise, you might be culturing, or you might be increasing the number of your cells, but they don't have the recombinant DNA, so you're just wasting your time. So these are the 4 important steps, techniques that are important for genetic engineering. So let's look at each one of these particularly. So when we talk about breaking and joining DNA molecules to form the recombinant DNA, you're essentially talking about making use of restriction enzyme and also making use of DNA ligase. So just remember that restriction enzyme are also called molecular scissors. So in this case, you will be cutting them at a specific restriction site. As we will be seeing later, you can produce overhangs, flaps, or what we call as sticky ends, while some will be producing blunt ends. On the other hand, once you have put together a DNA from one species and then combine this with the DNA of another species, you must be able to make this you must be able to put them together to seal the gap, to make it stable, and you make use of DNA ligase so that you will be able to really close the gaps. So that will be breaking and joining DNA molecule. On the second step, the suitable gene carrier, what we're talking about here is the vector. And the vector is that carrier. Particularly, the vectors are vehicles to carry particular DNA segments into a host cell as part of the cloning or recombinant DNA technique. There are 2 major usual or popular vectors. You have your viruses and you also have your plasmids. And more popular would be the plasmids, and as you have remembered from our video last time of how insulin is produced, the human insulin gene was inserted inside of a plasmid and then later placed inside of a bacteria. So these are two of the carriers that are very popular. The third technique or the third step would be the means of introducing the composite DNA into the host cell. So in this case, there are several ways, but for our SLG, we just made use of the gene gun, which is wherein the plasmid or the recombinant DNA is placed into a gold pellet, and just like a gun, it is fired into different cells, and the bullet or the gold bullet will pierce the membrane or pierce the cell wall, and then introducing or inserting the recombinant DNA into the cell. It's a little bit violent, but that's how it is done. And then there's another one, which is heat shock transformation. You just make use of heat in order to loosen the cell membrane, and then allowing the plasmid to probably insert into some of the holes that will be produced, making use of heat shock transformation. These are the two ways by which plasmid can be introduced to the host cell, which was introduced to us by our SLG. But do remember that there are several others, like making use of chemicals like calcium chloride and liposome, so that it will be able to let the plasmid, the recombinant DNA, go inside the cell. There is other physical means like microinjection, making use of a very tiny syringe, and electroporation, making use of electricity to make holes in the membranes of the cell. There's also vectors like viruses. You're very familiar with the life cycle, and also agrobacterium would be another way. So these are several means of introducing, but the SLG just mentions two of them, the gene gun and the heat shock transformation. Okay, the fourth one is method of selecting from a large population of cells a clone of recipients. So we mentioned that it is very important for us to be able to really determine which of our cells or the host cell actually contains the recombinant DNA, otherwise we're wasting resources. So one of the many ways would be making use of a plasmid containing the antibiotic resistant gene. So very straightforward ito. What you actually do is simply put all of your cells, you cultivate your cells or you culture your cells in a media inside of a petri dish that will be containing a certain amount of antibiotics. So you're very familiar, antibiotics will definitely kill the bacteria if it doesn't contain any resistance genes. So what we mean is that when the plasmid or the recombinant DNA is introduced, we're not very sure which cells would be containing that recombinant DNA, because it's not 100%. It's not 100% that you can be sure. So that when you put it inside a medium with antibiotic, since the recombinant plasmid will contain an antibiotic resistance gene, you would be very sure that the cell that contains the plasmid will be able to survive because they have the antibiotic resistant genes. It will produce a protein that will degrade the antibiotic, enabling it to live and to make a colony. So you would be expectation natin dito na kapag ka merong colony na nag-grow dyan in a medium with antibiotic, the recombinant plasmid is inside of that. And then of course bacteria without the plasmid, kasi they will not be containing the antibiotic resistant genes, you would be expecting them to die in that medium in the presence of the antibiotics. Okay, so that's it for the 4 steps of genetic engineering. Let's go straight to some other details that are very important. So of course, we have to remember that these are the very important actors for genetic engineering. So you have your DNA fragment or the gene of interest. You have your plasmid DNA, which is the vector. You have the restriction enzyme, the molecular scissors, and also the ligase that will seal the recombinant DNA. So these are the actors we have to remember. So as an overview, if this would be the region of, this would be the genome, this would be the gene of interest that we would like to insert inside of our vector. So making use of restriction enzyme, we're able to separate the gene of interest, and the restriction enzyme allows the formation of what we call as flaps or overhangs or sticky ends that will enable it to stick into the vector. Now, for the plasmid vector, we may also make use of the same restriction enzyme so that we will be assured that the gene of interest will be able to insert or to pair or to stick towards the vector or the plasmid, because you have the same overhangs, you have the same sticky ends, because you made use of the same restriction enzyme. And once you do that, they will stick together, and finally use ligase in order to permanently stick the gene of interest into the vector or the plasmid. And now you have your recombinant DNA. So that's the overview of how we do recombinant DNA technology. Now, one of the things we have to take note of, again, going back to the restriction enzymes, here are some details for that. So you have to, for example, EcoR1 restriction enzyme, you can see that the overhangs are formed, and this would be very specific nucleotide sequence, so take note of this one. So here are some example, HIND3, PS1, EcoRV. Notice that HIND3 and PS1 will produce overhangs, or what we call as sticky ends, while the EcoR5 or EcoRV will produce what we call as blunt ends. So there's a difference between them. Now, please take note, one of the important characteristic of the restriction enzyme is that it is palindromic. When we say palindromic, you will notice that they would have the same nucleotide sequence, but that will be in opposite direction. So for example, for the HIND3 restriction enzyme site, you will see AAGC double T. On the other hand, on the other side, you will see it as well. It's the same thing, but it's going to the other side. AAGC T and T. So this would be palindromic. So in order for us to be able to really determine if it's a restriction enzyme, it's restriction enzyme site, highly likely it will be if there will be palindromic, wherein opposite, the nucleotide sequence will be opposite of each other, the two strands of DNA. So take note of this one. So you see T, T, C, G, A, A. On the other side is double T, C, G, A as well. If you look at PST1, you have here C, T, G, C, A, G. If you look at the other strand going from the other side, C, T, G, C, A, G, it's the same. So it's called palindromic. Okay, here are some examples of those restriction enzymes, but no need to memorize this one. Just be familiar that there will be palindromic. You notice this one is palindromic. This one is also palindromic. This one is also palindromic. So that will be one of the characteristics that makes it highly likely to be a restriction enzyme site. Okay, take note of this one. Now, one of the skills that we have to learn is for us to be able to do restriction enzyme map problems, wherein we will be able to determine how many fragments are produced, and also take note of how many base pair are there for each of the fragments. So here's one question: how many fragments will be produced if the given plasmid is exposed to EcoR1? E is restriction site for EcoR1, while H is the restriction site for HIND3. So you have here on the left side diagram of a plasmid, the E being EcoR1 restriction sites, while the H will be HIND3 restriction site. So if the question would be, you will be exposing this to EcoR1, what will happen? Of course, there will be, it will cut the plasmid. Where will it cut? Since we have two sites for this, we will be expecting that a cut will happen here and a cut will happen on the other side. Alright, so the question is how many fragments are produced? You have two fragments here, fragment number 1 and also fragment number 2. So this is fragment number 1 because you have a cut over here and a cut over here. And then fragment number 2 will be over here because this would be, since the cut is here, it will go all the way over here up to this point. So you have two fragments. Now, if the question would be, what would be, how many bases per fragment, you just need to take note of what is given. So as you can can see here, from E to the other E, you have 100 base pairs. So definitely the first fragment will be 100 base pairs, while the other fragment, which will be very long over here, so you have a 200 to H, from H to another H is 200, H to another H is 100, H to the other H is 200, H to the E is 200. So you have to just add all of these things together, as you can see here. So 200 plus 200 plus 100 plus 200 plus 200 is equivalent to 900 base pairs. So that would be the answer to that particular question. Okay, how about making use of the other one? So how many fragments will be produced if the given plasmid is exposed to HIND3? So you have 1, 2, 3, 4 HIND3 restriction sites, so the E will not be cut because of the no echo R1 was placed here. So what do you do here? So you have to make a cut over here, another cut over here, another cut here, and another cut over here. So that will give us how many fragments? There will be 4 fragments. From H up to the other H is 1 fragment, H to the other H is second fragment, H to the other H is third fragment, and H to the other H will be the 4th fragment. So we have 4 fragments for this particular plasmid. Now the question would be, what would be the number of base pair? You just have to add all of them. So for the first fragment, you have H to the 1 to the E, you have 200, and then E to the other E is 100, E to the H 200. So you just have to add all of them, 200 plus 100 plus 200, and that is equivalent to 500 base pairs. For the second fragment, you have here 200, the third fragment is 100, and the fourth fragment is 200. So that is how you answer questions about what would be the number of base pairs for that. Okay, so that's very easy. Now let's look again at the overview of the what we call as the recombinant DNA technology. So you have here your foreign DNA, the desired gene, and then making use of the same restriction enzyme, you cut the plasmid as well, making sure that they have the same overhangs or the same what we call as the sticky ends. In addition to that, you have here putting them together and making use of DNA ligase to stick them together. And finally, we will transfect, or we will insert this into a bacteria and allow it to clone. So that will be the general ways by which the DNA or the DNA recombinant technology is done. Okay, some other terms we have to remember. Gene cloning is the process of synthesizing multiple copies of a particular DNA. Transformation or transfection, introduction of the foreign DNA into the cell. And third one is cloning. The transformed bacterial cell is grown in culture to multiply the recombinant plasmid.